Okay, thank you very much. And we are very happy to be here. And this is one thing I want to describe to you. And in my talk, I want to describe some of my work. It's about 1/5 of what we do in the lab. And also, I want to introduce some of the things that might be really interested to faculties here that we may have a chance to collaborate. So I will introduce some of the resources that we are building in this. And most of the work was done in New York when I was a faculty at Cornell Medical School and then NYU's Mount Sinai, but some of them I've continued at Peking University. So I want to introduce our work in making animal models for Parkinson's disease. To this audience, I don't need to go to very great details, but just a few sentences. Parkinson's disease is the second most common neurological diseases, so it's a big social economic burden to the society. Second, about 15%, 10 to 15%. Okay. Thank you. Yeah. About 15% are familial cases. Most of them are sporadic. Now, the cardinal phenotypes and symptoms of Parkinson's disease, there are several. One is bradykinesia, means people are very hard to initiate and sustain a movement. And second, there's resting tremor and rigidity, postural instability, and so on and so forth. Most of the patients are responding to levodopa treatment in a very good, sensitive way. So if we look at the genetic form of Parkinson's disease, there are several genes already being identified, and they fall into the category of autosomal dominant, autosomal recessive, and so on and so forth. And if we look at the cellular functions or dysfunctions of these involvements, they fall into all the places within a cell. It could be protein aggregation problem, it could be protein clearance problems, and so on and so forth. And now, recently, there are two genes isolated. Both of them are kinases. They are serine threonine kinases, and that is PINK1 and LRRK2. So in this huge list, I'll tell you about our work in LRRK2 most of the time. And if I have time, I'll introduce PINK1. If it's too long, I will just cut that part out. So LRRK2 gene, leucine-rich repeat kinase 2, the genetic inheritance is autosomal dominant in its mood. And the clinical manifestation is very, very typical idiopathic Parkinson's disease. Now, in studying this gene and the mutation, people found that mutations in LRRK2 not only is the highest prevalence in genetic form of Parkinson's disease, also it spills over to sporadic form big time. In some of the Jewish population, it's as high as 38%. In North Africa area, it's also as high as 38 to 40% of the sporadic cases that carry LRRK2 mutation. So that makes it a very, very important gene and protein to study. This protein is really interesting. It's a huge protein, RR domain for protein-protein interaction, WD40 domain for protein-protein interaction. And the fun thing is, in the middle, it has a GTPase domain and a kinase domain. Now, GTPases and kinases can regulate each other. That's not new. There are many cases. However, GTPase and kinase in one protein, one molecule, this is actually the very first case. The mutations on this gene fall all over the places. So we actually took some of the kinase mutations and GTPase mutations to make animal models. And that leads me to one of the main themes of my talk, that is animal models. To study these, it is, of course, we need to know the molecular biology and the protein chemistry of the signaling pathways. We need to know how the mutated protein or the absence of a protein is affecting subcellular functions or cellular functions. We also want to see that those mutations you can actually make an animal model that accurately recapitulates the disease. It sounds easy, but it's really, really not. In this category, in neurodegeneration, so far we have very few cases that we have recapitulated the cardinal phenotypes of neurodegeneration. That includes Alzheimer's disease, Huntington's disease, Parkinson's disease. So it's a big problem. The gold standard of good animal models, this is my definition. I think there are two aspects. First one, you need to recapitulate the cardinal phenotypes at every level, hopefully. The second one is you should not have those unwanted false phenotypes, which is usually there when you look at different animal models. Many years ago, when I was a postdoc at Rockefeller, William Yang and I, we were friends and PKU students together. So we actually invented a bacteria artificial chromosome, BAC transgenic methods. That one is really good to be used for animal modeling for diseases. What it does is really it expands the previous capacity for making transgenic animals from about 20 kb to nearly 400 kb long, which allows us to have a true gene in it. It has intron, exon boundaries, it has 5' and 3' regulatory regions, and so on and so forth. So we actually made this method at Rockefeller together, and we started to use this method for modeling diseases. And the very first one we did was Huntington's disease, really nice, and we continued, and I worked on Parkinson's disease. So to show you what we did with the mouse model for LRRK2, I want to show you our transgenic, back transgenic LRRK2 mouse. And these mice, they are born together as litter mates. And about 9 to 10 months, these ones carrying mutations, they started to move very slowly, and eventually they almost stopped moving. Oops. Now, the one that actually moves, that's the control. The one that does not carry the disease transgene. So seeing that, we were of course very happy. And next question would be...
So this is dominant?
This is dominant. Yeah, this is dominant disease.
Professor, are you wearing a cell phone maybe? You have a cell phone on you?
No, I don't have a cell phone.
Okay, go ahead. And there are options.
I don't have a cell phone.
Okay, okay, thank you.
Okay. Yeah, and now they start to move slowly, and they stop moving. That doesn't mean it's a problem with the dopaminergic system. So we did a very quick experiment simply to inject them with levodopa or apomorphine, see if we can rescue the dopamine deficit. And that's the result we have after about 30-40 minutes of injection. And this one is still the control, wild-type non-transgenic control. And these guys really start, oops, there's a mismatch of the signaling somehow. So the rescue is about 50-40%, which is very similar to human. So this, believe it or not, this is actually the very first mouse model that recapitulated age-dependent progressive loss of movement, and it's dopamine L-dopa sensitive. So, of course, we're very happy, and we want to use this one to study. And we looked at the dopamine transmission in these mice by microdialysis. So this is done on behaving, moving animals. And what we observed is the baseline of dopamine is not that big of a change, surprise to us. But then the dopamine transmission is defective by about 40-30%. And so you can see a clear dopamine transmission deficit in these mice compared to the non-transgenic litter mates. Now, if we look at the the histology, this is what we see, that in terms of the size of dopaminergic neurons, they have a clear atrophy. Now, if you count the numbers at 11 months, the number has a trend, but it's not statistically significant. The most obvious one, it doesn't even take a trained eye to see, is the degeneration of axons and dendrites. This is from age-matched, sex-matched transgenic animal with a disease gene. This is the wild type. And the brownish staining is the TH staining. So very clearly it goes through a quote-unquote degeneration from the dendrites and axons and then to the cell body. If we take a closer look at these axons or dendrites, this is what you see. You can find many typical axonal degeneration phenotypes, like those bulbs, like beads on a string type, the dysmorphic neurites, and it's very clear, and it's only in the transgenic animals. Now, interestingly, if we look at the hyperphosphorylated tau, that's also a pretty strong phenotype in the transgenic mouse. And then we went back to the human data and discovered that precisely the same mutation, the R141G mutation, in patients, that's the genotype that has a pretty strong tau phenotype, not other ones. So accidentally, we were right to recapitulate this phenotype with hyperphosphorylated tau as part of the pathology. And so those are all available to the community. I want to introduce to you the rat genetics aspect following the idea of animal modeling. So many years earlier than that, when I was working with PINK1 mutation, and PINK1 in mouse, it produced very little, if any, phenotypes. So I was a little bit disappointed, and that was the time I started to think about using rat as a genetic research organism. Rat has been really used very widely in research as well as pharmaceutical companies, but as a genetic tool, it's been lagging behind that of mice for a long time, by 15, 20 years. And I think there are reasons for us to really push rat back to the stage, because they are very useful features. Rats are much better for behavioral studies, especially those more sophisticated behaviors. Some of the electrophysiology are better done in rat, and it's just much more susceptible to disease insults for whatever reason. Especially in neurodegeneration, rat as a model is always easier to produce the phenotype if you use viral-mediated infection. So that's why my lab really jumped into that field. It nearly bankrupted my lab because it's so expensive, but very sort of satisfying to me that really now this field, rat, the genetics manipulation has been established, and reported our work. And then I want to show you what happens if you want to introduce the disease gene into rat. So this rat was born normal, and this is about, again, a year old. And if you put it into a cylinder, normally they would go around and they rear up to explore the environment. But this one, you can see it just wobbles, and it never stands up and explore the environment. So it drops to 0. Now, the same rat, if you, again, rescue that with apomorphine or L-DOPA, you can rescue, quite a bit of the mobility. Okay, Therefore, this is the very first rat genetic models, again, for Parkinson's disease, and it's a very nice one. We followed up with other disease genes for Parkinson's disease, and here's a list. If you look at the Taconic, I donated all of them to Taconic so that people in academic as well as pharmaceutical companies can use them. So all these rat models are from my lab. And interestingly, for those that we could not recapitulate the phenotype in mouse, now you have beautiful phenotypes. So following this one, when I returned to Peking University, now we succeeded in applying for a national platform building grant that will allow us to make many rat models for basic research as well as for medical study. And this is for scientists all over the world. So if you have your interested gene, let's say you are studying psychiatric disorders, and you come across with DISC1, SHANK3, neuroregulin, you say, aha. Be happy to do that. All you need is to write to me, and we will have a committee reviewing the priority of these requests, and we will make that. So we will ship the animals to you. So it's a very big resource to share with scientists all over the world. Now, after we have the good animal models, naturally we want to know a lot more about the mechanistic side of disease progression. So there are three questions we ask right away for LRRK2 because it's a kinase. So we want to ask what kinase cascade is activated. The second, does it change the protein phosphorylation profile? And the third one is, what are the direct substrates of LRRK2 if the kinase activity is the origin of pathogenesis? The first one, we adopted a profiling kinase cascades. The strategy is called ABPP, activity-based protein profiling, and the idea is as follows. So researchers have found that on the kinases, there are glycines that are very conserved. So we can design ATP analogs so that here is an R group and here is an ATP analog, and the R group is like this. Okay. Using this ATP analog, when a kinase is attacking its substrates, there's this reaction so that this kinase is attacked. And the kinases that are not active will not have this reaction. So this is why it's called active kinase ligand, activity-based. And we used this method to look at LRRK2, and to our surprise, we didn't get one. Other than itself, we have very little candidates coming up. And of course, we were suspicious of our technology, so we did it again. We went to the lab who invented this, and to our surprise, it's not. Therefore, contrary to the common belief that it's through a typical MAPK, MAPKKK, that sort of kinase signaling, the kinase cascade doesn't seem like it's the mainstream of LRRK2 pathogenesis pathway. Now, can we look at the substrates? It's a kinase. If it's transducing its signal from itself to another molecule, then we should be able to ask what the kinase substrates are, and maybe we can get a clue from that. Now, even the most competent biochemistry labs know that kinase substrates and kinase pairing, it's difficult because kinases are very efficient. They grab their substrates, phosphorylate them, kick them out. So it's very fast and transient. The normal way of making co-IP, for example, would not work. So we adopted a new way of what I called showcase method. And the essence of this method, biochemically, like this. So this is, the red color is a kinase structure. And if you look carefully, in the ATP pocket, this is ATP, the blue and cyan one. This is magnesium. And there is always a conserved amino acid whose side chain really points to the N6 position of ATP. It has to be there, and it has to be perfect in spacing. Cannot be too far, cannot be too close. And this one, Kevin Shokatz at UCSF calls it gatekeeper. It's a gatekeeper. Now, he's very smart. He says, look, if we engineer this amino acid and make it shorter, for example, change that into a glycine, basically chop off the side chain, that would lead to liberation of this whole space. What you do next is redesign your ATP so that your ATP in the N6 position, you have a bulky group. So it's an artificial ATP. That ATP will not get into normal kinase's pocket, so it's useless. It will get into your ATP, your kinase, engineered kinase, as a substrate. Of course, you can label your ATP with radioactivity or with other groups so that you can isolate that. Now, we actually followed that strategy and made LRRK2 with that gatekeeper mutation. And then we did experiments, and then we actually made a mouse out of that. And the idea is to really isolate LRRK2 substrates using this engineered mouse model specifically for that purpose. And I'm glad to show you that this is the control, and this is the bands that we are isolating individually from different cell fractions. And to our surprise again, there aren't that many. It's basically in the exonal transport that category. So it seems to me that LRRK2 is not a very upstream kinase that kicks in a whole cell death pathway and so on and so forth. It's very downstream to the exonal transport side, and somehow the dysregulation of LRRK2 leads to neurodegeneration in that direction. All right, seems I do have 6 minutes, and then I will quickly talk about another gene called PINK1.
Can I ask a question?
Sure, sure.
Is it a balance between the activity of a kinase and phosphatase, or is that there is a phosphatase involved also in this?
There has to be a phosphatases for most of the phosphoproteins. Then LRRK2 substrate, what phosphatases are responsible for removing phosphorylation, we still don't know. And phosphatase are much more promiscuous. There are a lot fewer phosphatases, and each one of them are responsible for a bunch of proteins.
And this mutation, does it affect the interactions of phosphatase? Do you have longer phosphorylation or constitutive phosphorylation?
That we don't know. That we don't know. Yeah, we don't even know which phosphatase is involved in that yet. Now, mitochondrial dysfunction has been discussed as one of those possible mechanisms for neurodegeneration for a long time. And what's missing is the causality. We don't know if it's a product, a consequence, or it's actually upstream. It's involved in many important cell functions. But then PINK1 gene for Parkinson's disease really addressed that question head on, because PINK1 is a nuclear encoded protein, but then it's shipped to mitochondria. So whatever it's done with PINK1 mutation, the first hit has to be in mitochondria. So it's the first case. And we are, of course, very interested in studying that. Now, PINK1 is a serine threonine kinase, and mutations are on those very conserved amino acids. So we made animal models, including Drosophila models. So the first question we asked was, well, okay, it's in mitochondria, great. Where in mitochondria? A simple question. So So we did a sucrose gradient experiment, and we found, interestingly, pink one almost never exists as a monomer. It's always together with those complexes. Now, what are those complexes? There are so many protein complexes, and the ones we suspect, of course, is the electron transport complex. And we know that it has complex 1, 2, 3, 4, and then each one of them consists of many subunits. For example, complex 1 has 46 or 47, depending on which group you ask. So we ask, well, with pink one mutation, do we see a deficit in these complex enzymatic activities or functions? We measure that by complex 1, 2, 3, 4 driven respiration. So this is what we saw. In a complex 1 and complex 4 driven respiration is greatly reduced, and we can isolate extracts and measure the activity, enzymatic activity of complex 1 and 4, and they have a matching deficit as the respiration experiment. Now, as a consequence of that, in the end, ATP synthesis with the mutant or pink one, absence of pink one, is reduced. So it all seems to fit. And then our next question is this reduction of ATP, does it have anything to do with protein clearance? Because in Parkinson's disease context, protein aggregation, the Lewy body, and the alpha-synuclein overexpression is a big problem. So we tested the proteasome activity with the absence of pink one. Yes, proteasome activity is reduced. Now, what does pink one do to the ETC complexes? So using the same strategy, we looked at the assembly of ETC complex, and we found that ETC complex assembly is much reduced with pink one knockout. So it seems that we are implying that phosphorylation or not of these ETC subunits affects its assembly, therefore affects its enzymatic activity as a complex, and therefore affects ATP production in the end. And we also looked at how the lack of pink one or mutant pink one affects alpha-synuclein aggregation, and we did that by cell culture experiment and filter trap. And without pink one, the protein aggregation is increased. Now, I do not want to leave you with a linear sort of thinking, oh great, phosphorylation decides the assembly, assembly decides the activity, activity defines ATP, and reduction of ATP causes proteasome deficit, therefore you have aggregation of alpha-synuclein. I don't think it's simple as that, but maybe this is one of the ways that pink one is causing a PD pathogenesis. And of course, we're asking pink one, what does it do to the ETC complex, and what is the phosphorylation substrate? So we used again this Kellogg-Showcat method, made mutations for pink one gatekeeper amino acid, made that into an animal, and look at that. And to our great satisfaction, these data are all coming out, and there's one paper last week published in Science. It's actually validating one of our discoveries. It is one of the 46 complex 1 subunits. It's really satisfying to see our results proved by another lab totally independently. So, and there are several of these bands. So we solved the question of some of them were actually now after the other bands. And what makes me feel difficult is I'm not sure whether they're all, the absence of phosphorylation of all of them together are making the problem, or some of them are more important than the others. And that's a difficult experiment to do. All right, to summarize, we have developed the methods for animal modeling for diseases. That's the first one is the back transgenic method. And the second one is the development of rat as a transgenic system that people can use. We have done investigation in multiple organisms and multiple approaches, and we have some of the discoveries that point to maybe their involvement in LRRK2 and the Pink1 mediated Parkinson's disease pathogenesis. So these are my people in the lab who are responsible for producing good work. And Bob Berg, Serge at Columbia University, they helped my lab when we started to do sterilology. And William Yang, this is my good friend who collaborated with me for the back methods, and we've been really talking to each other a lot. And Marie-Françoise taught us how to do behavioral studies. Thank you.
Thank you, for one more questions?
So phosphorylation seems to be very important. From my experience, I found that if you use, instead of using ATP as a substrate, you use ATP gamma S, then usually the phosphatases, the protein phosphatase, they cannot choke the gamma phosphate.
Right.
And remain phosphate. Did you try to do things like this?
No, I haven't.
This can be very useful to see the effect.
Is the phenotype, is it progressing one? That is, after the development of the akinesia
Right.
in your aids, if you continue to aid for another 2 months, will the phenotype will be more severe one?
Yeah, they really start moving, and we have to put food and water really in dishes right on the floor of the cage, otherwise they will just...
And the dopamine loss?
The dopamine cell loss in the rats, we're still counting that, but in the rat, it's the cell loss, the number, it's still mild.
Still mild, even if you wait a little bit more.
Yeah, still mild. But the dendrites in axonal retract is so obvious. It's just black and white.
And how is that related to Tom Schwartz's work? So Tom Schwartz showed that in the Pink1 mutation there's some difference in mobility of mitochondria and removal of this sick mitochondria, right?
Right. The mitophagy aspect. Yeah, I think it's gaining a lot of attention and interest. I think maybe it is involved in the removal of old mitochondria. Most of the Tom Schwartz lab is done in Drosophila. I don't think it's been replicated in mouse. Actually, we tried it and we couldn't replicate that in mouse. In Drosophila, interesting, Drosophila models for Parkinson's disease is really, really good for recessive and dominant. And so,
Better than mice.
Better than mice. Yeah, but rats, I have very good hope because we have done several models and they're all very successful. So, but mice are the recessive form. It's just so hard to have anything.
Okay, thank you very much.
Thank you all for staying with me. I'm going to move a little bit from the cellular and molecular level, which is over here, to the network and the behavioral level. And again, I'm going to speak about my main research all over the years have been done with primates, human and non-human primates, and especially with the MPTP model of Parkinson's disease and the human patient with idiopathic form. Although, again, as mentioned, at least 20% of our Ashkenazi Jews Parkinson's disease patients are LRRK2 positive, and we don't see big difference in the electrophysiology of LRRK2 positive patients versus patients that are not LRRK2, but it is still a mystery for us. Anyway, before going to Parkinson and our investigation, I would like to raise the general problem of how we can optimize our behavior. And the problem is that actually we are all agents that do an action on the world, and this action actually is changing the world a little bit. Okay, I'm now speaking to you. You might be interested. You might be looking at me, and so on. And actually, when we are now looking at this world, we get from the world a new state of the world and a reward. And this reward can be either positive or negative. If you are all sleeping already, it is a negative reward. If you are all interested and looking for the future, it will be a positive reward. And to make a long story short, we can say that the goal, the computational goal of this agent, and this agent can be a computer, but can be or a robot, but it can be you or your friend or your dog, is to maximize the future cumulative reward. And again, keep in mind that the reward can be either negative or positive. However, to achieve this goal is not an easy task, because first of all, it would be easy if there was a teacher that we can trust. And this teacher can tell us, go to the right side of the world, go to China, or stay in Jerusalem. And if there was a teacher that we take as a know-all teacher, then it would be very, very easy to say, this is the optimal behavioral policy. But there is no such teacher, and therefore we have to learn the optimal behavioral policy by exploration with the world, by taking our chance, going sometime here. And the problem is that the world is very, very large. So there is the curse of dimensionality. The world is too large to explore all possibility. It is noisy, and moreover, it's changing. So the rules that were correct today might be not correct tomorrow. And finally, the feedback is incomplete. That is, we don't get the feedback that tells us, this was the correct action, you've been 5 centimeters from the optimal target. We get reward, which can be a pleasure or non-pleasure, but nobody tells us how far from the target we've been. And moreover, it is delayed. That is, we are getting the feedback for our action in a kind of a temporal delay since the time that we've done it. So this is a very, it looks like a very easy problem, how we can learn without a teacher from exploration with the environment. And nevertheless, we can see that there are several problems that make this problem and not an art problem. Nevertheless, in computer science, especially in the field that we call today machine learning, a solution was found. And the solution was found mainly by dividing this agent into 2 parts. One part is the actor part, the main axis, that actually carry the behavioral policy. That is making the transition between the state, the current state, and the actions that we do. So, of course, this behavioral policy does not have to be deterministic one. It can be probabilistic one. It can change over time. But the point is that the actor is the coupling between the state and the action, and this is our behavioral policy. This is a very simple part of the agent. The more sophisticated part of the agent is the critic that all the time calculate the mismatch between prediction and reality. So we don't have a teacher, okay? So you came to this room and you have no idea if it's going to be an interesting or a very boring lecture. So your default assumption is 50-50, okay? This was your prediction. Now, 2 minutes later, you are a little bit clever. Hopefully, you think it is better than predicted, okay? So you continue to listen, okay? So what the critic is doing all the time is calculating the mismatch between prediction and reality. And if this mismatch is positive, reality is better than prediction, then we encourage the behavioral policy that was leading to this. If the mismatch is negative, reality is worse than prediction, we actually don't repeat this actions that were leading, or we change our behavioral policy. So we have actually a system that actually composed of a critic and an actor that eventually in machine learning we can show that enable us to achieve the computational goal, which is to maximize the cumulative future reward by interacting with the system and adjusting our behavioral policy despite the incomplete feedback and the noisy feature of the environment. So this is very nice in machine learning, and the nice feature actually is that we can now look back into our brain and we see that with machine learning we reinvented what we have already in our brain. Because to make a long story short, if you're looking, this is a coronal section in our brain, and you see over here the basal ganglia. And to make a long story short, we can say that if we are looking over here, this is the main axis of the basal ganglia, where all cortical field, the visual cortex encoding your visual input, the auditory cortex encoding your auditory input, the frontal cortex encoding your motivation, hippocampus encoding your memories, and so on. So all cortical field are projecting to the input stage of the basal ganglia, the striatum and the subthalamic nucleus. And then we are going through the internal circuits of the basal ganglia, external and internal segment of the globus pallidus, substantia nigra pars reticulata, and up to the motor centers of the brain, either in the brainstem or in the primary motor cortex, and we do action on the word. So this is the main axis, exactly like in machine learning. We have the state encoding over here, going through the basal ganglia, and coming to the motor center of behavior. But so if this is the actor, what is very unique about the basal ganglia, that we have more than one teacher. And what we can see is that the basal ganglia, of course, we have the dopamine teacher that we had already mentioned before, and there is a long background of studies showing that the dopamine neuron encode pleasure, but actually not pleasure, but pleasure prediction error, and so on and so on. So we have the dopamine teacher, but no, it is not the only teacher of the basal ganglia, because we have also the cholinergic teacher, the cholinergic interneuron of the striatum, the serotonin teacher coming from the raphe, and the histamine teacher. So we have multiple teachers in the basal ganglia, actually enabling us multi-objective optimization. And multi-objective optimization is better than maximization of cumulative reward. Because if we believe that the computational goal of the agent is to maximize the cumulative reward, actually we put ourselves only on a single axis. It could be pleasure or punishment. Everything is projected into a single axis. This might be correct for American. It is not correct for nations that are having a little bit of history, like over here, like in China. Okay, we would like to believe that we have more than one axis of emotion. Okay, and therefore we would like to stress this issue of multi-objective optimization. And actually, again, this is a kind of a new issue that you don't see too much in the field of reinforcement learning. And I would like to give you a kind of a short introduction to the question of multi-objective optimization. So let's say that you decide to go now and to buy a car, okay? And you know that actually when you buy a car, you have to think about two issues, okay, at least, okay? One is the cost of the car, okay? $10K, $100,000. And the second issue is the comfort, okay? How much pleasure you'll get from the car. And you are doing your homework and you understand that actually there is a correlation. The more you pay, the more comfort or the more pleasure you will get from the card, okay? But you do your homework and you understand that actually you can be somewhere, the best achievement will be over here. If you will pay this $100,000, you will get this Mercedes, okay? And there is nothing better. And if you are able to pay only $10,000, you will get this Volkswagen, okay? So you are doing your homework, okay? And then you are going to the car dealers, okay? And the car dealers, of course, give you this option. You say, this is the best for you. You will pay only $50,000, okay? And you will get this kind of comfort, okay? But you have done your homework, okay? You know what you have to do, okay? And you know that if you are ready to pay this $50,000, okay, then you should get this amount of comfort, okay? And if this amount of comfort is also good, is enough for you, then you should pay only $20,000. And eventually, you can be also over here in this point, okay? The major issue that what we've created now is the Pareto frontier, okay? Is the frontier that if you do multi-objective optimization, you should multi-objective optimization of maximizing the comfort and minimizing the cost, okay? This is the goal of now of your multi-objective optimization. You should be somewhere over here. Okay? It could be that you have constraint about this, the amount of money that you have in the bank, and so on and so on. But putting aside constraint, okay, there is nothing to say that B is better than A or worse than C. Okay? All these points on the Pareto frontiers are as legal or are Pareto optimal in the term of multi-objective optimization. Okay? And the question that actually what we'd like to say is that yes, this is the computational goal of the basal ganglia. Not only to maximize the pleasure, the computational goal of the basal ganglia is to maximize the gain and to minimize the cost. Exactly like we've done in the case of the car, with the car dealer. But the major issue actually is not that I'm claiming this is only the two axes, okay? Let's start with two axes, and then, of course, in the moment that we've done it from 1 to 2, we can go to 3, 4, and above. So the goal, the computational goal of the basal ganglia is to maximize the gain and to minimize the cost, okay? And this reinforcement actually can automatically lead, okay, and again, you can go to the mathematical details over here. This multi-objective optimization behavioral policy automatically lead to a softmax behavioral policy. So another step is to tell you what is a softmax behavioral policy. So what is the softmax behavioral policy? We already start. What is the behavioral policy? We have a state. For example, state 1, state 2, state 4, okay? And we have possible action to select for this state, okay? This is a behavioral policy. And let's say that we know that for state 1, if we select action 1, we'll get $80, okay? And for state 2, for state 1, if we select action 2, we'll get only $20, okay? We know, okay, this is our prediction for the situation, okay? And now the question, what is our behavioral policy, okay, that we can do when we are sitting in state 1, state 2, and state 3? We start with the gambling behavior, okay? You are now on vacation. You are in Las Vegas, okay? So you are over here, okay? You know that for this you will get $80, for this you will get $20, but you don't mind, okay? You gamble, okay? It is the time to explore, okay? So the gambling behavior is that you are in state 1, and nevertheless the probability to select action 1 or action 2 is equal, is 0.5, okay? This is a gambling policy. On the other side, the greedy policy, okay, if you're a dean, okay, or a big manager, okay, you will take only the greedy policy, and all the time you'll select A1, okay? So the probability for A1 is 1, and for A2 is 0. This is the greedy behavioral policy. Greedy is not a good policy, okay? Because if something is changing the world, okay, and suddenly over here you may get $200, you'll never explore this possibility. So in the middle, we have a matching, probability matching, okay, a behavioral policy, which say, okay, I expect $80 from here, $20 from here. In 80% of the cases, in the majority of the cases, I'll go to this, okay? In the minority of the cases, let's say 20% of the cases, I will go over here, okay? Because maybe life has been changing, okay? And we've said that actually the goal, or computational goal, is actually to enable us to live in a world that is noisy and changing, okay? And therefore, matching, probability matching is a very good behavioral policy. So we have these three behavioral policies, but actually we can get them in using a single behavioral policy. So sometimes we say, yes, sometimes for time we'd like to gamble, so for time to time we need to be greedy. Most of the time we'd like to do probability matching. What we found actually that softmax, this softmax that we say that we get from the multi-objective optimization, softmax behavioral policy enable like this. So what is a softmax behavioral policy? Softmax behavioral policy tells us that the probability that you'll do action 1 is equal to exponent with the Q, the value of this action, divided by the pseudo temperature. Okay? So it is, and then it normalizes all over action. So what is this pseudo temperature? Okay? This pseudo temperature is actually the simple way that you may think about it. If you have a lot of temperature, okay, in Las Vegas, okay, you gamble, okay? If you have a very low temperature, then you freeze, you don't move, okay? And this is exactly what we see over here, okay? So if we plot over here the probability that you select action J as a function of the Q, of the value of this action, then if the temperature is very, very high, and we put it in the softmax, we get the flat line. That is, no matter what is the Q, we select the gambling behavior, we select this action and this action with the same probability. If the temperature is very, very low, okay, you lose all your dopamine neuron, you are akinetic, okay, you don't move, you're in the greedy behavioral policy. And somewhere in the middle, we are on this green line, which is the probability matching. So we have one behavioral policy, okay, that actually enable us by adjusting the pseudo temperature to play with the different domain of these three different behavioral paradigms, from greedy to gambling. So this is very, very nice. And now we can go to the teacher of the basal ganglia. And what we are trying to say is that each one of the teacher, and again, I will not go to the reason why we put increased temperature for dopamine and 5-HT and decrease for cholinergic and histamine, okay? This is something that we have to do. But what is very important is that each one of the teacher can either increase or decrease the pseudo temperature. And each one of the teacher can also change the state to action association. It can change the efficacy of the synapse between the cortex and the striatum and make either to increase the association between a state and an action or to decrease the association between a state and an action. So this is very nice and this is very model and we are very happy. And the question if we can really prove or at least can provide some experimental support to it. So what we've done is for many years we are training our monkey to associate different visual cue either with future food, which is coming over here, future air puffs that the monkey don't like, and neutral cues that tell the monkey no food, no air puff. And you can see that the monkey, when we give only the food cue, the monkey has anticipatory lick and then lick. And then if we tell the monkey that is going to get air puff, he is blinking. And for the neutral cue, he is doing nothing, neither lick, neither blink. And we put multiple electrodes into different domains in the basal ganglia and do extracellular recording of action potential. And this is one example. This is 8 electrodes in the external segment of the globus pallidus. Each line is a spike. And we can detect this spike, and we can now record them and to see how the neural activity, this is in the globus pallidus, this is the cholinergic interneuron of the striatum, how this neural activity is related to the behavioral events that we see over here. And again, to make a long story short, okay, if we have this behavioral event with the cue and then outcome, and sometime, okay, we are using a probabilistic cue, so the outcome is omitted, what you may see is that the dopaminergic neuron that are shown over here have a very nice encoding of the cue and the outcome, okay? But the cholinergic interneuron don't encode the cue. We see that it does not matter if the monkey is getting the aversive cue in red, the neutral cue, or the reward cue, all the responses are similar. So this is in the cue period. But if we are going to the outcome period, for example, over here, when the outcome is omitted, you see that the dopamine neurons are not very good, okay? They don't make the distinction if I omit the food or the air puff, but the cholinergic interneuron do very nice encoding. So we have a multiple teacher, okay? This is one of the issue. It is not only the dopaminergic teacher, it is also the cholinergic teacher and so on, probably for the 5-HT and the histamine. And the second issue is that if we are looking at the main axis and using our ability to do cross-correlation analysis, we see that in the main axis, the neurons are independent of each other, where the teachers are strongly synchronized, and all the teachers give the same message. So this is our reinforcement, reinforcement-driven multi-objective optimization model. Of course, there are many, many questions regarding this model that we should also learn, but let's take it as a beginning. And now I'm really going to go fast, because we can go and see what is going on in the basal ganglia when we are losing our dopamine. And for this, in the monkey, we use the MPTP neurotoxin that kill all the dopaminergic neurons and actually give us the full spectrum of clinical symptoms, starting with akinesia, the lack of voluntary movement, but also rigidity and low-frequency tremor and postural deficits. And what we see that the uncorrelated activity in the normal monkey actually have been replaced by synchronous oscillation in the MPTP-treated monkey. So there is a huge amount of synchronous oscillation in the MPTP-treated monkey, and then we've done the next stage and say, if we inactivate, if we see this abnormal activity in the MPTP monkey, can we cure the Parkinsonian monkey by inactivation of the main axis? And we've done inactivation of the subthalamic nucleus. And this inactivation of the subthalamic nucleus actually ameliorates the akinesia, the rigidity, and the tremor of the monkey. And therefore, we've been very, very happy. And to make a long story short, this was the beginning. This was published in 1990, and this was the beginning of the deep brain stimulation method that have been done in more than 100,000 patients all over the world. We are very lucky to have a program of deep brain stimulation also in our place. And again, what we do is that we have to use electrophysiology in order to identify the subthalamic nucleus, which is a small target and not easy target, and we have to go over there. And therefore, actually, we can see that also in the human, we can see exactly the same oscillatory activity that we have described in the monkey. And the same power spectrum, and the major issue that we have seen is that the longer the oscillatory activity in the subthalamic nucleus over here, the better the outcome of the deep brain stimulation. So our conclusion is that actually this synchronous oscillation at the beta between 12 and 30 Hz is the neural signature of Parkinson's disease, and therefore we actually came back to the monkey and to ask ourselves, can we use this oscillatory activity in order to trigger stimulation? So not to stimulate all the time as we do now in our patient, but to use closed loop deep brain stimulation triggered by the abnormal activity. And the bottom line was, yes, this was very, very successful approach. So I'm in my last slide in summary. I believe that we have good evidence to say that the basal ganglia network, and the basal ganglia network for me is almost all the brain. So all cortical area projecting to the basal ganglia. So, but the diamond in this crown is the main circuit, so the striatum and the dopamine. So the basal ganglia network operate as critics actor network and do multi-objective optimization. This multi-objective optimization is enabled by the ability of the teacher of the basal ganglia not to change only the synaptic plasticity between the cortex and the striatum, but also to affect the excitability of neuron in the striatum, and so to provide a pseudo temperature signal to this neural network. And finally, basal ganglia disorder are caused by malfunction of one of the teachers, leading to abnormal activity in this main axis and inappropriate behavioral policy. And therefore, what I would like to suggest that if up to now we usually treat basal ganglia disorder by changing this kind, the critic. We give dopamine replacement therapy for Parkinson. We give neuroleptic, which are dopamine antagonist to schizophrenia, and so on. So we usually manipulate our SSRI for depression. So we usually manipulate this teacher with chemical manipulation. I believe that the future actually will be in exploiting the basal ganglia trick and actually use electrical stimulation of the main axis, and probably better with multi-objective optimization closed loop method in order to provide better treatment for our patient. Thank you.
Thank you very much, Hadai.
Why deep brain stimulation helps only a fraction of the patient?
No. Deep brain stimulation help every patient with idiopathic Parkinson's disease, okay? And it helps them as far as the disease, the human disease, is limited to the midbrain, to the dopaminergic system. In the minute that the disease spread outside of the midbrain, okay? And we have the Braak progression and so on, and we get Lewy body dementia and Parkinson's disease dementia, and we get diffuse cortical dysfunction, then the brain stimulation is not working. But for every patient with idiopathic Parkinson's disease, not with progressive supranuclear palsy, not with MSA, not with other diseases that mimic Parkinson, deep brain stimulation will be very, very effective.
How does the cerebellum fit into this?
Good question. So it depends. Of course, there is also the HPA, the hypophysa, pituitary axis, and so on. I'm omitted part, but actually, these days there is a lot of literature about input from the cross connection between the cerebellum and the striatum. Okay, so Peter Strick, with his virus injection, have shown very nice reciprocal connectivity between the cerebellum and the striatum. So from my point of view, the cerebellum is also part of this, okay? At least from the point of view of the basal ganglia, okay? So this.
And are motor strategies stored in the...
Okay, so again, clearly I overkilled, okay? It is not that all the brain is basal ganglia, okay? And there are still connection, let's say, at the level of CPGs, spinal cord, okay? one, very simple. And probably above the basal ganglia, these are the cortico-cortical connection, okay? So information is coming to the visual cortex and can go directly, or not directly, to the motor cortex without going down to the basal ganglia. So I think the major issue of the brain is this multiple level of hierarchy of connection between state and action. The basal ganglia are over there in order to enable implicit automatic learning. Okay? This is what the basal ganglia do. So declarative learning, it's probably done in the cortex-cortex pathway. Okay? But this implicit learning that we learn actually to control our posture and so on, this is done in the basal ganglia. And yes, this is stored, okay, from my point of view, in the efficacy of the corticostriatal synapse, that it is, can be easily modulated by, let's say, triple Ebbian mechanism with dopamine as a teacher over there, and there is a lot of literature regarding this.
Yeah, so just to follow up on that, what was brilliant. Mostly looking at the pseudo temperature, you're relating to, it appears to a very very well-defined point in time. You're without any prior experience. So you're relating to the information being treated as naive, and then a decision is made at that particular point. You're disregarding completely experience-dependent plasticity that defines automatic behaviors where we create habits. And I think stratum is also plays a major role in habit formation, and then in the modification based on habit formation. That's exactly what you were talking about now.
But this is exactly what I'm trying to say, that dopamine and other teachers have two roles. One role is exactly in this kind of plasticity, okay? This is well done. The plasticity and learning and adjusting behavior by modulation of the corticostriatal plasticity, yes, this is there and this is very important. But what I'm trying to explain is why I'm taking a Parkinsonian human patient or a monkey treated with MPTP. I inject dopamine agonist, okay? Apomorphine, ultra fast dopamine agonist, and in less than 1 minute, the monkey is awake and very active, okay? So 1 minute is not enough time to relearn everything. So what I'm trying to say, this teacher of the basal ganglia has two roles, okay? One is the modulation of the synaptic efficacy. Done. This is correct, and I'm not putting it aside. But they also have a very immediate role, which is the pseudo temperature, and this is change in the excitability, okay? So both are important.
Yeah, I have a conceptual question about the model. This assumes that start from agent, then action, then word, and reward. So I think this is a very good model for, I would say, for animals. But for human beings, in most cases, we found that people do something to change the word, which results in a reward toward like a social group include that agent, or even result in a reward toward others. It has nothing to do with the self. What do you think of this neural model can apply to such human behaviors?
Again, I'm a little bit too young to have such a broad model and understanding the world. But what I would like to say is, yes, for me, interaction, we as primates, okay, and human are primates, actually interaction with other part of the society is probably a very, very strong reward signal, okay? And moreover, we can speak about our internal rewards. For example, if I have done something, okay, and it was just correct, I found a great hypothesis, okay? It is not that I got a chocolate, but somehow the completeness, so I have a kind of internal reward. So from my point of view, yes, I still believe that we are biological creature like everybody else, okay? And we are over there in order to maximize the amount of gene that we are moving to the next generation, okay? And biology has to play with us somehow to enforce us to find the optimal behavioral policy, okay? And it is not easy because we are aware of our action, and it is a kind of a closed loop between our reflection for what we've done and so on and so on. But yet, nevertheless, I think this is also a proper model if you agree with the kind of social interaction are rewarding as well, and internal reward model. And we can discuss it. But it is a little bit more philosophy. If I'll be able to explain everything up to human, it is good enough for me.
Thank you very much.